Today's topic is gene interaction. In Mendelism, we have seen that only a single gene is responsible or expression of a single trait. But what is gene interaction? When more than two genes are responsible for the one single character or trait or factor is known as gene interaction. That is, two genes are interacting among them to produce a definite character or a definite trait. But Mendelism, we have seen that how a single gene was responsible for a single character. But this gene interaction topic is a completely different from that of the Mendelism. What is gene interaction? Okay, when two or more different gene, I have told you that two or more different gene means non-allelic gene. You know the meaning of allelic. Allelic means that is an alternative pair of a gene. Okay, when these alternative pair of gene, for example, is for the, let's say, for height, the allelic pair are capital T small t, and for color of the, or you can say for the shape of the seed, the alleles are capital R small r. These capital T small t and these capital R small r are two non-allelic gene. Okay, that is known as non-allelic. Allelic means single gene, two alternative forms is allelic, and another gene alleles, and these alleles are known as the non-allelic form. As you can see here, this capital C small c and these pp are the non-allelic genes. Okay, what I was trying to convey. So when two or more different genes, that is non-allelic, influence the outcome of single trait, as I have told you that gene interaction means the interaction of two genes for the outcome of a single trait, but here the two different genes are the non-allelic gene. So what do you mean by that? Here these capital C small c and these capital P small p will work together for the single trait, that is known as gene interaction. And this gene interaction was proposed by Sir William Bateson and Reginald Punnett. Okay, these two scientists proposed this gene interaction concept. And where did they studied or observed? They saw under the sweet pea plant, that is Lathyrus odontrus, and they observed, they made certain experiments and proved the fact of gene interaction. What did they made? What are the following experiments which they conducted to show that what is gene interaction? So let's discuss. What he did, first he took a normal pure breeding, or you can say true breeding purple flower. What do you mean by true breeding? Means pure, nothing hybrid is not involved, that is known as pure breeding. So when he took pure purple flower and crossed with or hybridized with white flower in the F1 generation, he saw that all the flowers were purple color. Okay, next when he took the F2 generation, he saw that the flower was 3 shows 3 is to 1 ratio, that is 3 for purple and 1 for white flower. Okay, next when he took two varieties of purple flower in the F1 generation, he saw that purple flower was formed. Sorry, here two varieties of white flower, very sorry to say, that is it is white in color. Okay, when he took two varieties of white flower, two varieties means from two different genes, then he saw that in F1 generation all the flowers was purple color, and in the F2 generation he saw the ratio, that is 9:7. How is that possible? In the first case, when he crossed or hybridized purple flower and white flower, he got purple flower. But when he took two varieties of white flower, he got purple flower. How is that possible? Because of the gene interaction. How is it took place? Let's see. Suppose for the genotype, that is capital C, and for say let the genotype capital C or capital C small c. You can see for the genotype capital C, enzyme A is responsible. Okay, so for the colorless precursor enzyme, when enzyme A was given and a colorless intermediate was formed. Okay, next another enzyme B for which the genotype capital P and capital P small p is responsible. So you can see that is the presence of capital P. When enzyme B was given, a purple color, that is anthocyanin, was present. So by this experiment you can conclude that when both the capital C and the capital P was present, then purple color flower was present. So you can see here, this is one allele, this is another allele. These are the two non-allelic genes. So when these two non-allelic genes interacted among themselves, they produced one trait that or color that is known as the purple pigment. Here you can see here C represents the purple color producing allele and is dominant to small c, that is white flower, and capital P is also purple color producing allele and it is dominant over small p, that is white. So here in this condition, when only single capital C is present, then it produced a colorless intermediate, that is no purple color was seen. Here also you can see when the when the capital P genotype was produced, then only the purple pigment was produced. So when both this genotype or both this capital, these two will be present, then only the purple pigment will be present. For a better understanding, let's see the chart. Okay, let's see the crossing over. So when he took two white color flower plant, here only I have mentioned, this is by mistake I have written purple flower, but when he took two varieties of white flower in the F1 generation. So next what he did, he took two white flower, that is capital C, capital C, you can see capital C for purple, small c, small p for white. Okay, so when he took white flower plant, here why, you must be wondering why this capital C is present but it is white flower plant. As I have already told you that if single capital C is present, then it cannot produce a purple color. Okay, here also you can see, here only capital P is present, no C is present, no capital C is present, that's why white color flower is produced. By this you must be clear that if both the capital C and capital P is present, then only the white color, then only the purple color will be produced, otherwise white color. Here also only single capital C and capital P is absent, that was white color flower plant, and here also only capital P is present, that's why white flower plant is present. So when these two was crossed, as a result of which, due to crossing over, what happened, the capital C and capital P both are present together. That's why the flower which was produced is all purple. By now you must be clear why this point, what happened in this point, why purple flower will be formed. Because these capital C, when these capital C and capital P interacted among themselves, that is when gene interaction was seen, then the purple color flower was formed. Okay. Next what happened, when these F1 hybrid, that is the progeny of this F1 hybrid, was self-fertilized. Self-fertilized means it was crossed with itself only. Then we got the following F2 generation. What happened? We got 9:7 ratio. What is 9:7 ratio? That is 9 purple and 7 white. You can see wherever the capital C and capital P will be present, then the purple color flower will be produced. As you can see in this that is in this Punnett square, that is here in the first, when capital C, capital P was crossed with each other, then you can see here the purple color flower is produced because both are present. Here also, as a result, you can count here 1, 2, 3, 4, 5, okay, 6, 1, let's say 1, 2, 3, 4, okay, 5, 6, 7, 8 and 9. Okay, by this you can know what is 9. Why it is white in color here? Because only capital C is present, that's why it is white. So if you count all this, this is 9. So we got the ratio, that is 9:7. By now you must be clear why we got this ratio 9:7, and when we crossed two varieties of white flower, we got purple flower because of this gene interaction. Okay, so actually what is gene interaction? This is the phenomenon of epistasis. What is epistasis? We will be discussing in detail in the next class, but now what is difference between epistasis and dominance? Okay, epistasis means masking of or suppressing another allele, that is known as epistasis. But what is the major difference between epistasis and dominance? Because dominance also means the suppressing another gene, okay, another allele, but here the major difference is that in the dominant character the allelic suppression takes place. That is, if a gene is present, suppose for tallness two alleles are responsible, that is capital T and small t. Here the capital T is dominant over small t. This is the example of dominance. But in case of epistasis, that is, two non-allelic genes will be present and one allelic gene will be suppressing another allelic gene. For example, here if capital T small t will be one allelic gene, another will be let us say capital P and small p will be present. Okay, then these capital T of one gene will suppress this capital P and small p of another gene. So that's why it is known as non-allelic suppression and dominance is known as allelic suppression. This is very important point to be noted down. Another difference is that it involves a single pair of alleles. Okay, as you have seen here, only single pair of alleles are involved, but here two pair of alleles are involved, that is known as epistasis. Okay, here gene suppresses the expression of its allele, here the gene suppresses the expression of non-allele, as I have already told you. Next, the effect of recessive allele is suppressed, as I have already told you, that is the capital T will suppress the small t. But here the epistatic allele suppress the effect of both dominant and recessive non-allele. I have already told you that these capital T will suppress both capital P and small p in case of epistasis. So this is the major difference between dominance and epistasis. In dominant it is allelic, epistasis is non-allelic suppression. Dominance will suppress its allele only, its recessive allele only to be very precise, but epistasis will suppress both dominant and recessive non-allele, because here the capital T will suppress both capital P and small p. Next, the effect is only due to dominant allele in case of dominance, but here epistasis can be due to dominant and as well as recessive allele. And when we will discuss the chapter epistasis, we will see how many types of epistasis and what are the that is the genotypic and the phenotypic ratio we get from the crosses, because there are different types of epistasis which we will be discussing in the upcoming video. So that's all for today. Hope you are clear. If you have any doubt, you can please contact in the comment section. And that's all for today. In the next upcoming video, we will be discussing about what is epistasis, what are the types of epistasis, and etc. So thank you and have a nice day.